About Golgi complex, we will discuss today. So, Golgi complex is a continuation part of our last class here. In last class, we discussed about endoplasmic reticulum. Two types of endoplasmic reticulums are there. One is rough and another one is smooth endoplasmic reticulum. Here, about Golgi complex is another organelle or a part in your ninth class textbook of biology. And what is that here? Camillo Golgi. Camillo Golgi was the scientist. I will just explain it. Camillo Golgi. Camillo Golgi. First observed that this organ in the year 1898 using a very old, that means compound or microscope he used, and its a fine structure could be observed only under an electronic microscope. In electronic microscope, this kind of fine structure we can observe, but in normal compound what we use in schools, that will give you only just a small region like that. But in electron microscope you can see like this is the picture in electron from electron microscope only. And Camillo Golgi first discovered this Golgi complex. It is one cell organelle, cell part, and then this name was came after the scientist only. And here, if you see why it is called as complex, means like apartment, first apartment, second floor, third floor, fourth, fifth, sixth, like that floors are there. So that's why it is called as a complex. And the next one, this organelle is made up of several membranes are there here. These membranes will create a sac like. Sac means here a bag like. See here, like a bag it is there, like a bag it is there. Inside we can store some objects like that, it is there. So these sac like structures will around which fluid like vesicles are bound. These are fluid like vesicles which are called as vesicles. Understood? These are called as Golgi vesicles. These are called as Golgi vesicles. These are present in sac-like structures. And again, it is filled with fluid. The proteins and other substances produced in ribosomes, we will discuss that also, will reach us into this Golgi body through some vesicles. And here these substances are changed a little. And then that means here these ribosomes will change into little variation will be seen. And then these the substances which are changed, in one sense the function of Golgi bodies is pack of various substances before they are transported into other parts of the cell. That means these are changed into other forms before they are reaching into the main cell. And from here the substances are sent to cell membrane. So cell membrane will be there for our cell. So here Golgi complex will be there like this. And then these are after completion of changed structure, they will send into the cell membrane. That you need to remember. And from there to another organelle called as a lysosome. That means they just produce these organelles and chemicals, and then they will send into lysosome structure. After reaching the cell membrane, these substances are secreted from the cell, and sometimes even to regenerate or repair the membrane. These will help in repairing the cell membrane. Whenever we are getting injuries, then these cells will be helping in case of from injuries, accidents, or anywhere. Our skin will be cured again. New skin will be getting. You will think sometime that new skin will be getting only due to this kind of Golgi vesicles. And after that, lysosomes are there. So lysosomes, what are these lysosomes? Most important function is there here. Lysosomes, here you can see these are called as lysosomes. What is the main function here? All the lysosomes are formed from the Golgi complex, which we discussed now only. And one of the facts that troubled the scientist for a long time was that certain enzymes are present in the cell and that have the ability to destroy almost all the structures. That means here some enzymes are there in our body, in our cell, that will destroy very dangerous microorganisms or anything coming into our body. And this will destroy almost the structures in the cell, but it won't damage. It will just destroy, but it didn't damage the cell itself. So this puzzle was solved when lysosomes was discovered, as that these are very tiny particles here, if you see, visible in the cytoplasm, and it was found that they contained destructive enzymes. Inside that, some destructive enzymes are present here, and these destructive enzymes will normally do not come in contact with the rest of the cell. They will be present in some part of the cell. The materials that are needed to destroy are transported to the lysosomes. That means here, to kill any foreign object, for example coronavirus is there, sometimes it will enter into our body, and then these lysosomes you need to thank. Lysosomes are killing this coronavirus in our body. But if more virus enters into our body, lysosomes also cannot do anything. Like that, if any thieves are coming to state, to our state, maximum 1 lakh thieves are came, how come, so that police cannot do anything. Similarly, many coronavirus enters into our body. These lysosomes also cannot do anything. But lysosomes get them digested at sometimes, and these lysosomes sometimes what it will do, whenever you are drinking impure water or eating waste food on the roadside food here, so these are the microorganisms which are entering into our body, that will enter into the lysosomes, and lysosomes will kill this microorganism. Usually this is a function, but sometimes what will happen here, these lysosomes will burst itself due to kill the microorganisms or any other harmful organism. That's why they are called as, that means they are killing by themselves to kill microorganisms. That's why they are called as a suicidal bags. Suicidal bags of the cell. Suicidal bags of the cell. So that's why it is called as the suicidal bags of the cell. In some cases, what will happen here, if that microorganism is unable to kill, then lysosome itself will kill itself. So that when lysosome is killed itself, then the microorganism will also die. So that process is called as suicidal bags of the cell. Then we will discuss about mitochondria. Mitochondria, before going to that, we will have an experiment also. What is that here? We need to make first onion peel, and we need to add a small onion peel we need to take out. That means onion will be there, so onion peel will be there, so that peel we need to take out. Like this will be there, the peel, and on that we need to add Janus green bee solution. Janus green bee solution. So then this Janus green bee solution, how we need to add here, 200 mg in 100 ml of water. So 200 mg means a small tablet like structure, that much only, a little pinch in 100 ml of water. That means half of the glass of the water is will be made. And then next we need to take one a glass, that is otherwise called as a watch glass. So what is watch glass here? Now we have taken onion peel, and after that solution we prepared, and then next watch glass will be there like this. That watch glass we need to take, and whatever we poured made the solution, we need to pour to fill this watch glass. After that we need to put this onion peel for half an hour. Understood? That means for 30 minutes we need to put in that only the peel, and after that the slide we need to take, and then this small piece of onion we need to put over the slide, and again with the help of water we need to wash it very slowly, such that extra water will be removed off by the chemical. And after that we need to cover with the help of cover slip, and then if we see through microscope we can see this structure. Then what we can see here, mitochondria we can see. Mitochondria is a cell organelle. Here we will just observe that same like this only we will observe. And now we will discuss about the cytoplasm, the mitochondria here, which is there in your textbook. And mitochondria is most important. Why it is most important here? It is otherwise called as powerhouse of the cell. It is called as powerhouse of the cell. Why it is called as powerhouse of the cell? It provides the power to our body. That means energy will be given to our body. If you are walking, reading, writing, eating, so these all will take a huge energy. That energy will be supplied by this mitochondria. So you must thank all the time for mitochondria for giving energy. Now I am teaching means here only due to have mitochondria energy I am teaching now. So what are these mitochondria here? Mitochondria parts are there here. Just you see here, mitochondrial DNA, ribosomes, small parts are called as ribosomes, and this is called as matrix. Empty region is called as matrix. Two membranes are there here. One is outer, one is inner membrane, and between the two membrane space is called as intermembrane space. And next one here, yeah, this L-shaped structures are called as cristae, and these are called as ATP synthase enzymes. This name is not needed. Synthase, ATP synthase, SYNTHASE. So these are the main parts here. Horseshoe shaped will be there, this mitochondria, and wavy like patterns are called as cristae, and inside that membrane, that means inside the empty space, is called as a matrix, and they have round spherical structures called as ribosomes, and in that coiled structure is called as a DNA, and two membranes will be there, inside that space will be there, and two membranes is called as outer membrane and inner membrane. These are the parts here, and what it is important now we will see here. Mitochondria are very small, to say where they are very small, and sometimes spherical or cylindrical. Sometimes spherical or cylindrical will be there. Generally mitochondria will have a size of 2 to 8 micron in size, and after that that means the length, and this side if you see it is nearly 0.5 micron. Understood? This side it is 2 to 8 micron, and after that it is nearly 150 times smaller than the nucleus. That means in a nucleus 150 mitochondria can fit easily. So in one nucleus 150 mitochondria can fit easily. That means 150 times smaller than the nucleus, and these are about nearly 100 to 150 mitochondria present in each cell, ranging in a plant cell or animal cell, any cell. It consists of nearly 100 to 150 mitochondria. When seen under microscope, compound microscope, the mitochondria will appear like a oval shape, like this oval shape. It will appear like oval shape. That you need to remember in your exam. Usually will be asked here. So oval shape is most important, or sometimes cylindrical shape. Like this cylindrical shape will be there, and the diagram you can see here, it has a unique structure here. And if we discuss about much information when we have known from the electronic microscope, it is made up of double membrane. First, double membrane means here two membranes, outer membrane and inner membrane, and that means interior will protrudes and folds into structures called as cristae, and the space between the structures is called as matrix. Already I discussed now. And mitochondria is responsible for cellular respiration. That you need to remember. Cellular respiration, it is most important, by which energy will be derived from the cell to do work. Daily work will be doing yoga or otherwise exercises, walking, eating, running, talking, whatever you use in mobile with your hands. For that, energy will be getting through mitochondria. That's why it is called as powerhouses. Powerhouses of the cell. So powerhouse of the cell is called as mitochondria, and the suicidal bags of the cell is called as lysosomes. Kitchen house of the cell is called as chloroplast. These you need to remember, most important for this ninth class. Not only ninth class, in 10th class also these topics will be there. And ribosomes here, if you see, small ribosomes will be there here. Can you see these ribosomes? Very small ribosomes are there here, which are found in a cytoplasm of the cell, and these are called as that means small granule like structures are called as ribosomes, and they are formed by RNA process, a process called as RNA process. It is formed, that means through RNA proteins. Through RNA proteins it is formed. You need to remember a small topic only. 1 mark question will be there in your exams here, ninth exams. You need to remember, and they are of two types here: free ribosomes and attached ribosomes. So we discussed about endoplasmic reticulum here, smooth endoplasmic reticulum and rough endoplasmic reticulum. Rough endoplasmic reticulum will have attached ribosomes, and smooth endoplasmic reticulum is smooth. Why? Because they don't have any ribosomes. That means freely scattered here and there in the cell, and these ribosomes are main function of these ribosomes are here. They help in protein synthesis. That you need to remember. These help in protein synthesis. And again, if we discuss here, one activity is there. What is that activity means here? To observe chloroplast, kitchen house of the cell. Chloroplast here, there is a rheoleaf activity. So when a lab starts, you can do this in your school. So we need to take one rheoleaf here. Rheoleaf will be found in water. So nearby waters we can get that. And then just we need to take a small peel. That small peel we need to put under or on a slide. The small piece we need to put over slide. And just we need to observe under microscope. Then you can see chloroplast. That is one activity. And one more activity here. You can see One more activity here you can see is that algae activity is to see. Some areas will be getting algae, that means here it green in color substance here. If you see under any tab, so green color material will be formed. In rainy season it is much, so under the tap, wherever water is there or nearby the tanks or old buildings, algae will be formed. That we need to take and we need to put on a cover slip, then we will find algae materials. And one more is there algae, that means about chloroplast. That will be there. And chloroplast we will discuss in another class. It is a kitchen house of the cell. As of today, whatever diagrams are completed, you need to draw in your book, and whatever notes I given now, important notes like mitochondria are present 100 to 115 SL. So these notes you need to complete in your book, and after that you need to revise. If you want to make the video in YouTube only, you will be having a slow motion and a fast speed, so that you need to apply for revision purposes.